Imagine this, you finish a heavy lunch at 2 p.m. By 6 p.m., you haven't eaten anything, but your brain is still working. Your heart is beating, and your blood glucose remains almost normal. How? Where did the glucose come from? Did your body magically create it? Or did it store it somewhere? Today, you will understand the body's glucose bank, that is the glycogen. And trust me, if you understand this lecture, glycogenesis, glycogenolysis, and hormonal regulation and glycogen storage diseases will become incredibly easy for you. Before we begin, why should MBBS students care about the glycogen? Because this chapter concept connects the multiple important topics, that is diabetes mellitus, hypoglycemia, liver physiology, muscle physiology, hormonal regulation, and glycogen storage diseases. And in your university exams, explain glycogenesis, differentiate liver and muscle glycogen, write the functions of the glycogen. These are some favorite exam questions. So don't memorize, let's understand. The first question is obvious. What exactly is the glycogen? Think of the glycogen as a storage form of glucose in the animals. Here's a simple analogy. Imagine glucose is a cash in your pocket and you don't carry all your money in your pocket. Most of it stays safely in your bank account. Similarly, our body doesn't keep excess glucose freely circulating in blood. Instead, it converts glucose into glycogen and store it in for future use. Now, another question is, where is glycogen stored? According to this, mainly two places, that's the liver and muscles. Liver is 6 to 8%, and in muscles 1 to 2%. Now, students often ask, if liver has only a small mass compared to muscles, why is it so important? We will answer that shortly. Inside the cell, glycogen is present in the cytosol, but it doesn't float around randomly and it packed into the glycogen granules. Imagine a rice stored in a sacks instead of scattering grains everywhere. That's exactly the glycogen granules do. They keep glucose organized and ready whenever it needed. Now let's understand why glycogen exists. Here's the function. First, maintains the blood glucose levels. Blood glucose rises, the liver says, I don't need all this glucose right now. So it converts excess glucose into the glycogen. Several hours later, blood glucose starts falling. Now the liver breaks the glycogen and releases the glucose back into the blood. That's why blood glucose remains fairly stable between the meals. If liver glycogen is deficient, the patient develops fasting hypoglycemia. Remember this, it becomes the important in glycogen storage diseases. Second function is liver glycogen stores increase during the well-fed stage, and during fasting these stores decreases. Very logical that the fed state means storing and fasting means the spending. Think of it exactly like depositing and withdrawing money from the bank account. Next function, that the muscle glycogen act as a fuel reserve. Imagine you are climbing a stairs or running for a bus. Muscles suddenly need ATP. Do they wait for glucose from liver? No. They immediately use their own glycogen. That's why the muscle glycogen is like an emergency battery. This is one of the most commonly misunderstood concept, that the you might think fat stores much more energy. So why doesn't the body use fat all the time? Excellent question. That let's answer it. There are some reasons that the glycogen can be mobilized very rapidly. And imagine there's the emergency. Would you rather withdraw money instantly from your saving account or sell your house? Obviously, the saving account. Glycogen is your saving account and fat is your fixed deposit. Glycogen can produce energy even without the oxygen. This is the extremely important during sprinting. Oxygen supply becomes insufficient, fat oxidation cannot continue efficiently, but glycogen still produces ATP through the aerobic glycolysis. That's why athletes depends heavily on the muscle glycogen. The brain depends mainly on the glucose, and brain cells continuously require glucose. Liver glycogen helps maintain this supply, especially between the meals. Fat mobilization is slow, it needs the oxygen, and fat cannot directly provide glucose. Therefore, when the body urgently needs glucose, glycogen is always preferred. Glycogen is a saving account and fat is the fixed deposit. Now we know why glycogen is important. But how is it made? This process is called as the glycogenesis. The glycogenesis is the synthesis of glycogen from the glucose. It occurs in the cytosol and it requires ATP, UTP, and the glucose. Now let's build the glycogen step by step. Step first, the formation of the UDP glucose. Everything starts with glucose in muscle. Enzyme is hexokinase, and in liver, glucokinase. These enzymes convert glucose into glucose 6 phosphate, and the enzyme is phosphoglucomutase. It converts the glucose 6 phosphate into glucose 1 phosphate. Now comes the favorite university question that glucose 1 phosphate combines to UTP and forms the UDP glucose using the enzyme UDP glucose pyrophosphate phosphorylase. And don't think UDP as energy, think of it as a carrier that delivers activated glucose to the growing glycogen chain. Second step is, here's the interesting question: can glycogen synthesize start making glycogen from nothing? No, it always needs a starter, a primer. Earlier, scientists believed that the primer always comes from the existing glycogen. But what if no glycogen is present? Nature has a brilliant solution. Protein called the glycogenin act as a starter, and only then can glycogen synthesis continues. Here is the one exam tip that the glycogenin starts and glycogen synthase extends. Step third is the glycogen synthase. Now main enzyme enters the glycogen synthase. The enzyme transfers the glucose from the UDP glucose to the growing glycogen chain. It forms alpha 1,4 glycosidic bonds and repeats this clearly. Glycogen synthase forms only alpha 1,4 glycosidic bonds. It cannot make branches, and students frequently confuse this. Step 4 is the branching enzyme. If the glycogen remains in the straight chain, breaking it down would be the very slow. So nature creates the branches. The enzyme is branching enzyme, and that is the glucosyl alpha 4 6 transferase, and it transfers the small block about the 5 to 8 glucose residues to the another position. This forms the alpha 1 6 glycosidic bond. Now glycogen becomes highly branched. Why is branching is useful? Because more branches ends mean faster glycogen synthesis and faster glycogen breakdown whenever the body needs energy. Then we will see about the next part. In the previous part we have seen the body stores excess glucose as the glycogen, but here's the question: what the use of the storing money in the bank if you can never withdraw it? Exactly the same applies to the glycogen, and when blood glucose falls or muscles suddenly need energy, glycogen must be broken down quickly. That process is called as the glycogenolysis. Here is the overall reaction for the glycogenesis. Requires the 2 ATP, and here is the glucose plus glucose at the N plus 1 plus 2 ADP. 2 ATP is utilized and 1 is required for phosphorylation of the glucose, while the other is needed for conversion of UDP to UTP. Here is the whole reaction for the glycogenesis. Here are the glycogen synthase enzyme, and here is the transferase enzyme glucosyl alpha 4 6 transferase. Here is the elongation by the glycogen synthase, and here is the branching enzyme by the glucosyl 4 6 transferase. Here is the next glycogenolysis. Let's begin with definition. Glycogenolysis is the degradation of stored glycogen into the glucose containing molecules. It mainly occurs in liver and skeletal muscles like the glycogenesis. It also takes place in the cytosol. Can glycogenesis simply run in reverse to break glycogen? No. Body uses completely different set of enzymes. Why? Because using separate enzymes also allows the body to regulate synthesis and break down independently. This is the favorite Viva question. The glycogenesis and glycogenolysis are not reversible pathways. Remember the glycogen structure from part 1. It has alpha 1,4 and alpha 1,6 glycosidic bonds. Alpha 1,6 glycosidic bonds form the branches. Here both types of bonds must be removed. Now let's meet the star enzyme, that is the Glycogen Phosphorylase in step 1. This enzyme removes glucose one molecule at a time and form from the non-reducing ends of the glycogen. Notice something interesting, that it does not use water. Instead, it uses the inorganic phosphate. This process is called as phosphorylasis. And instead of this releasing free glucose, glycogen phosphorylase releases the glucose 1 phosphate. Now here's a catch. Does this enzyme continue until glycogen disappears? No, it stops when it reaches 4 glucose residues before the branch point. This remaining branched structure is called as the limit dextrin. Why can't glycogen phosphorylase continue? Because it cannot break alpha 1,6 bonds. It only breaks alpha 1,4 bonds. This is one of the most frequently asked university questions. In the step 2, debranching enzyme is there. Now we have a problem that the branches are blocking further breakdown. Who solved this? Debranching enzyme. Here's something very interesting. The debranching enzyme is the bifunctional enzyme. Let's understand them one by one. Activity first, that the transferase activity. The enzyme first takes the 3 glucose residues from the branch, it transfers them onto the nearby straight chain. Notice carefully that no glucose is released here and simply shifts of the glucose molecules. And in activity second, the alpha 1,6 glucosidase activity. Only one glucose molecule remains attached through alpha 1,6 bond, and second activity of the enzyme breaks this bond. This time, free glucose is released and not a glucose 1 phosphate, free glucose. So students commonly confuse this. The phosphorylase, glucose 1 phosphate, and debranching enzyme, free glucose. Only about 8 to 10% of the glucose released during the glycogenolysis is a free glucose because branch points are relatively few. In the third step, we will see the formation of glucose 6 phosphate. Now we have a glucose 1 phosphate. Can the cell use it directly? No. First, the enzyme phosphoglucomutase converts it into the glucose 6 phosphate. Then, the enzyme here now becomes an important difference between liver and muscle. Suppose the glycogen broken into the liver contains the enzyme glucose 6 phosphatase. Then, this enzyme removes the phosphate group. So, glucose 6 phosphate becomes the free glucose, which enters the bloodstream and maintains the blood glucose. Now think about the skeletal muscle. Does the muscle have glucose 6 phosphatase? No. Therefore, the muscle cannot release glucose into the blood. Instead of glucose 6 phosphate immediately enters the glycolysis to produce ATP for muscle contraction. Here's the memory trick, that liver loves blood and muscle loves itself. Liver shares the glucose and muscle uses its own glucose. Suddenly, it has some confusion. Another pathway is the lysosomal degradation. This is the pathway. These are the limit dextrins act on the debranching enzyme and forms free glucoses, and further action of phosphorylase forms the glucose 6 phosphatase and glucose. So far we have discussed a major pathway, but a tiny amount of glycogen is also degraded inside the lysosomes. The enzyme involved is acid maltase, and here the glycogen accumulates inside the lysosomes. This causes the Pompe disease, the glycogen storage disease type 2. Pompe disease, the acid maltase deficiency, and lysosomal glycogen accumulation. This is a classic MBBS exam question. Next we will see about the next part. So far we have learned the two opposite pathways, and one stores the glucose as a glycogen and other breaks down the glycogen into glucose. But here's the very important question, can both pathways run at the full speed at the same time? Imagine filling water tank while opening the drain at the bottom. Does that make sense? Absolutely not. Your body is so far smarter than that. Today we were going to learn how the body decides whether to store glycogen or break it down. This is one of the most important and most frequently MBBS exam question. The regulation of the glycogen metabolism is essential for the maintaining The normal blood glucose levels, only two enzymes decide the fate of the glycogen. That are the glycogen synthase and glycogen phosphorylase. That glycogen synthase builds the glycogen and glycogen phosphorylase breaks the glycogen. If glycogen synthase is active, storage occurs. If glycogen phosphorylase is active, breakdown occurs. The body controls these enzymes in three mechanisms, that are allosteric regulation, hormonal regulation, and calcium regulation. First one is the allosteric regulation. Before talking about hormones, let's understand the fastest type of the regulation, allosteric regulation. What does allosteric means? It means the molecule bind to the enzyme at the site other than the active site, changing the enzyme's activity. Think of it like a little light switch. The enzyme is already present. The body simply it's turn on or off. Well-fed stage. Imagine you have just eaten a heavy meal. Blood glucose is high, and inside the cell, glucose 6 phosphate levels also rise. Now ask yourself, should the body store glucose or break glycogen? Obviously, store it. So, glucose 6 phosphate allosterically activates the glycogen synthase. More glycogen is formed. Here's inhibition of the glycogen breakdown at the same time. High ATP and high glucose 6 phosphate signal that the cell already has enough energy. Therefore, inhibit the glycogen phosphorylase. No need to waste the glycogen. Here's the role of the free glucose in the liver. Here, free glucose itself acts as an allosteric inhibitor for the glycogen phosphorylase. Why? Because if the glucose is already high, liver glycogen breakdown stops automatically. What happens during exercise? Now imagine you are running and ATP gets consumed rapidly, AMP increases, and low ATP at a high AMP tell the muscle. And we need energy immediately. AMP directly activates the muscle glycogen and muscle glycogen phosphorylase, and that results into the rapid glycogen breakdown and rapid ATP production. Now let's move to the most important part, hormones. Almost every university asks this pathway instead of memorizing. Let's understand the logic. There are two situations. Blood glucose is low, and which hormones is increased? Glucagon and epinephrine. There's the simple job that increase the blood glucose. How? By breaking the glycogen. Binding the here's the breakdown pathway for the glucagon and epinephrine. These hormones bind to the receptors and on the cell membrane. Binding activates the formation of cyclic AMP. Remember, cyclic AMP is the secondary messenger, and second messenger, and hormone is the first messenger. Cyclic AMP activates and protein kinase, that is PKA. Now everything starts happening very quickly. PKA activates phosphorylase kinase, and phosphorylase kinase then phosphorylates the glycogen phosphorylase. Once phosphorylated, the glycogen phosphorylase become active and results the massive glycogen breakdown. But something else happens simultaneously. The PKA also phosphorylates the glycogen synthase. What happens now? It becomes inactive. Why? Because it would be foolish to synthesize glycogen while simultaneously breaking it. Here's the synthesis pathway. Now imagine you have just eaten, the blood glucose rises and insulin is released. The goal is to exactly opposite, store glucose, not break it. Insulin activates and protein phosphatases, unlike kinases and phosphatase, remove the phosphate groups, and these phosphatases remove phosphate from the glycogen synthase. Once dephosphorylated, glycogen synthase become active and glycogen synthesis begins. The same phosphatases remove the phosphate from the glycogen phosphorylases, and phosphorylase kinase both become inactive, so glycogen breakdown stops. And one final regulator is the calcium. Why would the calcium regulate the glycogen? Think about the muscle contraction. Whenever the muscle contracts, calcium is released from the sarcoplasmic reticulum. Now the muscle suddenly needs ATP and calcium binds to the calmodulin. The complex directly activates the phosphorylase kinase even without cyclic AMP. As a result, glycogen phosphorylase becomes active, more glycogen is broken down, and more ATP is produced. This beautifully matches the energy production with the muscle contraction. During exercise, calcium AMP work together and rapidly stimulates the glycogen breakdown in the muscle. Now here is the one concept. Suppose the glycogenesis and the glycogenolysis both occur continuously at equal rates. What happens? ATP get wasted and no useful work is done. This is called as the futile cycle. Normally, the body minimizes futile cycles by regulating enzymes through protein kinases and protein phosphatases. These enzymes act as molecular on-off switches, ensuring the only one pathway predominates the accordingly to the body's metabolic needs. Let's discuss about the glycogen storage diseases. These are inherited metabolic disorders caused by the defects in enzymes responsible for the glycogen synthesis or degradation. Because of these enzymes, either glycogen cannot be synthesized properly or it accumulates abnormally inside the tissues. Which organs are affected normally? The liver, skeletal muscle, and heart. Let's discuss the disease that the von Gersh disease type 1. Now let's study most important glycogen storage disease for MBBS. This is the glycogen storage type 1. It is inherited as a autosomal recessive disorder. The defective enzyme is glucose 6 phosphatase. Glucose 6 phosphatase, it is the deficient here. And let's understand the symptom at time. Normally during fasting, the liver breaks down glycogen and forms the glucose 6 phosphate and finally convert it into the free glucose. But in the von Gersh disease, the last step cannot occur. Therefore, the glucose cannot released into blood and result the severe fasting hypoglycemia. Second one is the lactic acidosis. Here's the another interesting question. If the glucose 6 phosphate cannot become glucose, where does it go? It accumulated in glucose 6 phosphate enters the glycolysis and glycolysis produces pyruvate which converted into lactate. So blood lactate level rises. This causes the lactic acidosis. Third one is hyperlipidemia. Now the body still needs energy. Since the glucose cannot be released, it starts mobilizing the fats. Fat breakdown increases. As a result, the blood contains more free fatty acid, triglycerides, and ketone bodies. This produces hyperlipidemia. Hyperuricemia, fourth one, this is one of the most confusing symptoms. Let's simplify it. When glucose 6 phosphate accumulates, some of it enters in the HMP shunt and this pathway produces more ribose 5 phosphate. Ribose 5 phosphate increases the production of purine nucleotides and when excess purine are broken down, they produce the uric acid. Therefore, blood uric acid rises and this causes the hyperuricemia. And high uric acid may eventually lead to gout, causing painful joints. Here we have studied the whole glycogen metabolism. So, if you like it, share, like, and subscribe. And congratulations, you have now completed your entire chapter of glycogen metabolism from the glycogen structure and functions and glycogenesis, glycogenolysis and regulation and glycogen storage disease.